This video reviews the process of DNA extraction for high throughput sequencing. In other words, the DNA extraction for the purpose of next-gen sequencing of the entire microbiome of individual beetles. We're using the QIAGEN DNeasy blood and tissue DNA extraction kit, and you should be following along with the protocol that we've prepared on this process. We're also assuming that you begin viewing this video after having performed surface sterilization of each of the beetles that you plan to do an extraction from, and that those individual beetles are now in sterile, individually sterile microfuge tubes, which you can see in front of you. So here you can see individual microfuge tubes with beetles in them, individual beetles that have already been surface sterilized. You begin this process by pipetting 180 microliters of the buffer that's labeled ATL from the QIAGEN kit. So you're going to put 180 microliters of ATL buffer into a single microfuge tube containing the beetle that you are going to do your extraction from. As you're doing these procedures, I recommend that you use a fresh pipette tip every time you're going into a new sample. Even though you might be doing more than one sample at the same time, getting the same reagent, to avoid cross-contamination, use a fresh pipette tip each time you go into a sample. Then obtain a sterile microfuge pestle and use that pestle to thoroughly grind your bean beetle in that ATL buffer. You may need to press on the sides of the tube to grind the beetle properly. You will have fragments in there, but the idea is to get a nice slurry in that buffer of the material from the beetle. The liquid will be cloudy once you've done this grinding properly. Your instructor may direct you to save the used pestle. They can be washed and re-autoclaved and then reused. The next step is to add 20 microliters of proteinase K, then put some gloves on, because the next step requires that you use a buffer. This buffer is called buffer AL, and buffer AL contains guanidine hydrochloride, which is toxic, and you don't want to get it on your skin. This particular reagent reacts badly with bleach, and so if you spill it, do not use bleach to wipe it up. You want to use laboratory detergent and water if you spill it. But again, this next step, immediately after adding 20 microliters of proteinase K, is to add 200 microliters of buffer AL, then mix thoroughly by vortexing. You should actually vortex for 10 seconds, and then move your sample to a dry bath incubator. After vortexing, then incubate your microfuge tube with the ground up beetle that contains the buffers ATL and proteinase K and buffer AL in a dry bath at 56 degrees Celsius for 10 minutes. While you're waiting for your sample to incubate for 10 minutes at 56 degrees Celsius, you should obtain from your instructor a DNeasy mini spin column. These are columns that come with the QIAGEN kit. They're in a shrink wrap package. Each one is individually wrapped. Go ahead and unwrap the column. The column itself is actually placed in, or comes placed in, a small collection tube. So pull the column out. That's what I'm pointing to right now. That's the column at the top. And the collection tube below is just for collecting material that flows through the column. Mark the column so you know that your sample is in that column. After your sample has incubated for 10 minutes at 56 degrees Celsius, you will add 200 microliters of reagent grade 96 to 100% ethanol to each sample, and then mix thoroughly by vortexing for 10 seconds. Now the entire contents of your extraction, of your sample, needs to be transferred using a micropipetter to the column, to the DNeasy mini spin column. When you're doing this transfer, make sure that the mini spin column is in the collection tube that it came in. And you want to get as much of the liquid from your sample into that column as possible. It's all right to transfer some beetle parts with that. That's not a problem at all. Picking up some parts, beetle parts, won't interfere with the extraction process. With the mini spin column in its collection tube, transfer those spin columns and collection tubes to a centrifuge where you will centrifuge for 1 minute at 10,000 Gs. Remember, you want to balance the tubes, so they need to be completely directly across from each other, and make sure you secure the lid on the centrifuge as well. Once the centrifuge has stopped spinning, carefully remove your tubes, and you will see that there's a liquid that's collected in the collection tube of each sample. That liquid is waste and may be disposed of. The DNA that you're trying to purify is in the column that's on the top of that collection tube. So you're going to be removing the collection tube, disposing of the collection tube with the liquid that's in the collection tube, saving the column and putting that column in a new collection tube for the next step. Once you've put the spin column in a new collection tube, you will add 500 microliters of buffer AW1 to the mini spin column. Then centrifuge for 1 minute, again at 10,000 Gs. When that spin is finished, again there will be a liquid in the collection tube at the bottom. That liquid is waste. You're going to take the spin column and put it in a new collection tube and dispose of the flow through. And now you will add 500 microliters of buffer AW2 to each mini spin column. Again, the mini spin column needs to be in a collection tube before you add the buffer to the column. Once you've added the 500 microliters of buffer AW2, then spin the columns for 3 minutes at 20,000 Gs, or the highest speed you can get from your centrifuge. This centrifugation step is a drying step. You're attempting to dry the mini spin column membrane to remove all the ethanol that was being washed through. So if there's any residual liquid on the tip of the column, you may need to spin it a second time. Carefully remove the column from the tube, the flow-through tube that contains waste. Dispose of the flow-through tube, and now place the column on a sterile microfuge tube, a microfuge tube that has a cap on it. Make sure that that microfuge tube is labeled with your sample name, your group name. Label it DNA. So you're placing the mini spin column onto a sterile microfuge tube that is labeled with your sample name, and it's also labeled DNA. Then you're going to add to that column 100 microliters of molecular grade, sterile, nuclease-free water. You're going to add that water directly onto the membrane itself. Be careful not to let the pipette tip touch the membrane, but squirt that 100 microliters of water onto the top, and then just let it sit for 1 minute at room temperature. Then carefully load the microfuge tube with the column in it into the centrifuge. Again, balance your tubes in the centrifuge. It's important to orient the tubes, that is, to orient the microfuge tubes, so that the caps of the microfuge tubes are sideways. This way, when you spin the tube, those caps will not be accidentally sheared off. Look at the picture in your protocol so that you see exactly how they should be oriented. Spin the tube for 1 minute at 16,000 Gs, and the liquid that comes out of the mini spin column now is going to be water that contains your purified DNA. So this last step contains the DNA that you're interested in. Do not discard this DNA. After that spin of 1 minute, then you can remove the column from the microfuge tube, dispose of the column, close your microfuge tube, seal it with a piece of parafilm, and provide this tube to your instructor. This is your purified DNA.